We spent time looking at the outside of plants, leaves, stems, roots, flowers. But just like you can't understand how a car works by only looking at the paint job, we need to look inside plants. The study of plant internal structure is called plant anatomy. And here's how plants are organized. Cells form the basic units. These cells organize into tissues, and tissues organize into organs. Now here's something important. Within flowering plants, monocots and dicots are anatomically different on the inside. The internal structures also show adaptations to diverse environments. A desert plant looks different inside than a rainforest plant. And different organs within the same plant show structural differences too. A root's internal structure is quite different from a stem structure. Before we dive into specific organs, let's understand how plants organize their internal tissues. On the basis of structure and location, there are 3 types of tissue systems. Think of them as 3 departments in the plant. First is the epidermal tissue system. This forms the outermost covering of the whole plant body. It's like the plant's skin. The second system is the ground tissue. This makes up the bulk of the plant body. It's all the tissues except the epidermis and vascular bundles. And third is the vascular tissue system, which consists of xylem and phloem. This is the plant's plumbing system that transports water, minerals, and food. Let's start from the outside and work our way in. The epidermal tissue system comprises 3 things: epidermal cells, stomata, and the epidermal appendages like trichomes and hairs. The epidermis itself is usually just one layer of elongated cells that are compactly arranged to form a continuous protective layer. Think of it as shrink wrap around the plant. Now, epidermal cells are parenchymatous with just a small amount of cytoplasm lining the cell wall and a large vacuole. Plants have this clever waterproofing trick. The outside of the epidermis is often covered with a waxy thick layer called the cuticle. This prevents water loss. But here's an exception. The cuticle is absent in roots because roots need to absorb water, not prevent its loss. Stomata are really important structures in the leaf epidermis. They regulate 2 key processes, transpiration and gaseous exchange. Let me tell you about their structure. Each stoma has 2 bean-shaped cells called guard cells that create an opening between them. That's the stomatal pore. In grasses, these guard cells are dumbbell-shaped instead. The guard cells have an interesting wall structure. The outer walls away from the pore are thin, but the inner walls facing the pore are highly thickened. This matters for how they open and close. Guard cells are unique because they possess chloroplasts, which most epidermal cells don't have. They use these to regulate the opening and closing of the pore. Sometimes you'll see a few epidermal cells near the guard cells that look different. We call these subsidiary cells. When we talk about the complete structure, the pore, the guard cells, and the surrounding subsidiary cells, we call this whole functional unit the stomatal apparatus. The epidermis bears different types of hairs depending on where you are in the plant. Root hairs are unicellular elongations of the epidermal cells. They're like fingers reaching out into the soil to absorb water and minerals. On stems, the epidermal hairs are called trichomes, and these are usually multicellular in the shoot system. Trichomes come in many forms. They may be branched or unbranched. They may be soft or stiff. Some are even secretory, producing various substances. But their main function is helping prevent water loss due to transpiration. They create a layer that reduces air movement right at the surface. Now that we've covered the outer coating, let's look at what fills most of the plant body. The ground tissue is defined as all tissues except the epidermis and vascular bundles. It consists of 3 types of simple tissues: Parenchyma, Collenchyma, and Sclerenchyma. In primary stems and roots, you'll find parenchymatous cells in the cortex, pericycle, pith, and medullary rays. In leaves, the ground tissue has a special name and function. It's called mesophyll, and it consists of thin-walled cells containing chloroplasts. This is where photosynthesis happens. We've seen the protective layer and the filling. Now for the plumbing system. The vascular system consists of 2 complex tissues, the phloem and the xylem. When xylem and phloem occur together, we call them vascular bundles. Here's a crucial difference. In dicot stems, cambium is present between the phloem and xylem. Because of this cambium, these bundles can form secondary xylem and phloem tissues, so we call them open vascular bundles. They're open for growth. In monocots, the vascular bundles have no cambium. Since they don't form secondary tissues, they're called closed bundles. Vascular bundles can be arranged in different ways. When xylem and phloem are arranged in an alternate manner along different radii, we call this radial arrangement. You'll see this pattern in roots. In contrast, when xylem and phloem are jointly situated along the same radius, it's called conjoint arrangement. Such bundles are common in stems and leaves. In these conjoint bundles, the phloem is located only on the outer side of the xylem. Always remember this positioning. Theory is good, but let's see how this actually looks in real organs. We'll start with a dicot root, working from outside to inside. The outermost layer is called the epidermis. Many cells of the epidermis protrude outward as unicellular root hairs. Next comes the cortex, which consists of several layers of thin-walled parenchyma cells with intercellular spaces between them. The innermost layer of the cortex is called the endodermis, and this is really important. It's a single layer of barrel-shaped cells with no intercellular spaces. The endodermis has this amazing feature called Casparian strips. These are depositions of suberin, a waxy, water-impermeable material, on both the tangential and radial walls of the cells. These strips act like gaskets, controlling what enters the central part of the root. Next to the endodermis lies the pericycle, a few layers of thick-walled parenchymatous cells. The pericycle has important functions. It's where lateral roots originate, and during secondary growth, the vascular cambium also starts from these cells. So it has meristematic potential. Now we reach the vascular bundles. In dicot roots, there are usually 2 to 4 xylem and phloem patches. Later in development, a cambium ring develops between the xylem and phloem. The parenchymatous cells lying between the xylem and phloem are called conjunctive tissue. The pith in dicot roots is small or inconspicuous. All the tissues on the inner side of the endodermis, that's the pericycle, vascular bundles, and pith together, constitute what we call the stele. The anatomy of the monocot root is similar to the dicot root in many respects. It has the same basic layers: epidermis, cortex, endodermis, pericycle, vascular bundles, and pith. The overall organization is the same. But here are the key differences. First, count the xylem bundles. Monocots usually have more than 6. We call this the polyarch condition, where poly means many. Remember, dicots typically have 2 to 4. Second, the pith is large and well-developed in monocots, whereas it was small in dicots. Third, and this is important, monocotyledonous roots do not undergo any secondary growth. Let's come up from roots and look at stems. In a young dicot stem, the epidermis is the outermost protective layer, covered with a thin cuticle. It may bear trichomes and a few stomata. Below this, the cortex consists of multiple layers arranged in 3 subzones. The outer subzone is the hypodermis, made of a few layers of collenchymatous cells. This provides mechanical strength to the young stem. The cortical layers below the hypodermis consist of rounded, thin-walled parenchymatous cells with conspicuous intercellular spaces. This is the general cortex. The innermost layer of the cortex is the endodermis. In stems, the endodermal cells are rich in starch grains, so this layer is also called the starch sheath. The pericycle is present on the inner side of the endodermis and appears as semilunar patches of Sclerenchyma above the phloem. Here's a diagnostic feature. A large number of vascular bundles are arranged in a ring. This ring arrangement of vascular bundles is the characteristic feature of dicot stems. If you see a ring, you know it's a dicot. Each bundle is conjoint, meaning xylem and phloem are together. They're open, meaning cambium is present. And the protoxylem is endarch, meaning it's positioned toward the center. In between the vascular bundles, you'll find a few layers of radially placed parenchymatous cells. These constitute the medullary rays, They provide radial connections. The central portion of the stem is occupied by the pith, which consists of a large number of rounded parenchymatous cells with large intercellular spaces. Monocot stems look completely different inside. First, the hypodermis is sclerenchymatous, not collenchymatous like in dicots. Second, and this is the most obvious difference, the vascular bundles are scattered throughout the stem, not arranged in a ring. Each bundle is surrounded by its own sclerenchymatous bundle sheath. There's a large, conspicuous parenchymatous ground tissue filling the stem. It's not differentiated into cortex and pith like in dicots. The vascular bundles are conjoint but closed, meaning no cambium is present. Notice that peripheral bundles are generally smaller than the centrally located ones. Two other unique features. Phloem parenchyma is absent, and you'll find water-containing cavities within the vascular bundles. From the stem, let's move out to where photosynthesis happens, the leaves. A vertical section through a dicot leaf shows three main parts: epidermis, mesophyll, and vascular system. The epidermis covers both the upper surface, called adaxial epidermis, and the lower surface, called abaxial epidermis. Both have a conspicuous cuticle. Here's an interesting asymmetry. The abaxial or lower epidermis generally bears more stomata than the adaxial or upper epidermis. In fact, the upper surface may even lack stomata completely. This makes sense because the lower surface is more shaded and loses less water. The tissue between the two epidermal layers is called the mesophyll. It possesses chloroplasts and carries out photosynthesis. The mesophyll is made up of parenchyma, but it has two distinct types of cells. The palisade parenchyma is placed on the adaxial or upper side. It's made up of elongated cells that are arranged vertically and parallel to each other, like a palisade fence. The spongy parenchyma is quite different. It consists of oval or round cells that are loosely arranged. It's situated below the palisade cells and extends all the way to the lower epidermis. There are numerous large spaces and air cavities between these cells. These air spaces allow for gas exchange with the stomata. The vascular system in leaves includes vascular bundles, which you can see in the veins and the midrib. The size of the vascular bundles depends on the size of the veins. In dicot leaves with reticulate venation, the veins vary in thickness, so bundle sizes also vary. Each vascular bundle is surrounded by a layer of thick-walled bundle sheath cells. This provides protection and support to the conducting tissues. The anatomy of the monocot or isobilateral leaf is similar to the dicot leaf in many ways, but shows some characteristic differences. First, in an isobilateral leaf, stomata are present on both the upper and lower surfaces of the epidermis, not just the lower surface. Second, the mesophyll is not differentiated into palisade and spongy parenchyma. It's more uniform throughout. Grasses have these special cells called bulliform cells. These are certain adaxial epidermal cells along the veins that are large, empty, and colorless. They're like water sensors for the leaf. When the bulliform cells have absorbed water and are turgid, the leaf surface stays flat and exposed. But when they're flaccid due to water stress, they deflate and make the leaves curl inwards. This is a survival trick to minimize water loss. Another feature, the parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles throughout the leaf. Now that we've seen each organ type, let's directly compare monocots and dicots. In roots, both have the same basic organization with epidermis, cortex, endodermis, pericycle, and vascular bundles. But look at the differences. Dicot roots have 2 to 4 xylem bundles, a small or inconspicuous pith, and they undergo secondary growth with cambium formation. Monocot roots have more than 6 xylem bundles. That's the polyarch condition. The pith is large and well developed, and they do not undergo any secondary growth. There's no cambium formation. These differences make identification pretty straightforward once you count the xylem bundles and check the pith size. Stem anatomy shows even more dramatic differences. In dicot stems, the vascular bundles are arranged in a ring. This is the diagnostic feature. The bundles are open with cambium present, allowing secondary growth. The hypodermis is collenchymatous for flexible support, and the ground tissue is clearly differentiated into cortex and pith. Monocot stems look completely different. The vascular bundles are scattered throughout the stem, not in a ring. The bundles are closed with no cambium, so no secondary growth occurs. The hypodermis is sclerenchymatous, providing rigid support. There's a large, undifferentiated ground tissue, and each bundle has its own sclerenchymatous sheath. If you cut a corn stem and a sunflower stem, the difference is immediately visible. Leaf anatomy also shows clear differences. Dicot leaves are dorsiventral, meaning the upper and lower surfaces are different. Stomata are mainly on the lower surface. The mesophyll is differentiated into palisade parenchyma on top and spongy parenchyma below. The reticulate venation pattern means vascular bundle sizes vary. Monocot leaves are isobilateral. Both surfaces are similar. Stomata are present on both upper and lower surfaces. The mesophyll is not differentiated. It's uniform throughout. Parallel venation results in similar-sized vascular bundles. And grasses have those special bulliform cells that allow the leaf to curl during water stress. Let me give you a quick identification guide. For roots, count the xylem bundles. 2 to 4 means dicot. More than 6 means monocot. Check the pith size, small in dicots, large in monocots. Remember that roots always have radial arrangement of vascular tissues. For stems, look at how the bundles are arranged. A ring pattern means dicot. Scattered means monocot. Check whether cambium is present. If yes, it's an open bundle and you're looking at a dicot. If no cambium, it's a closed bundle from a monocot. For leaves, check where the stomata are located. Mainly on the lower surface indicates a dicot. On both surfaces indicates a monocot. Examine the mesophyll. Differentiated into distinct palisade and spongy layers means dicot. Uniform throughout means monocot. And the venation pattern is a dead giveaway. Reticulate is dicot. Parallel is monocot. Let's test what you've learned. Suppose you observe a transverse section under the microscope and you see these features. Conjoint, scattered vascular bundles surrounded by bundle sheaths, and phloem parenchyma is absent. What would you identify this as? Think about it. Scattered bundles immediately suggest monocot. The bundle sheaths confirm it. An absent phloem parenchyma? That's a specific monocot stem feature. So this is a monocot stem. Here's a practical tip for when you cut a transverse section of a young stem and observe it under the microscope. First, look at the overall arrangement of vascular bundles. Is it a ring or scattered? This alone often gives you the answer. Then check whether there's cambium between the xylem and phloem. Note what type of cells make up the hypodermis, Collenchyma or Sclerenchyma, and check for bundle sheaths around individual bundles. These 4 observations will let you confidently identify whether you're looking at a monocot or dicot stem. Let's bring this all together. Plants organize their tissues into 3 major systems. The epidermal system provides protection and includes structures like stomata for gas exchange and trichomes to reduce water loss. The ground tissue system forms the bulk of the plant body and consists of parenchyma, collenchyma, and sclerenchyma. The vascular tissue system handles transport, with xylem and phloem forming vascular bundles. Remember, the structure and function of these tissues depend on their location in the plant body. Monocots and dicots show consistent anatomical differences across all organs. In roots, it's about bundle number, pith size, and whether secondary growth occurs. In stems, look for ring versus scattered arrangement, open versus closed bundles, and hypodermis type. In leaves, check stomatal distribution, whether the mesophyll is differentiated, and the venation pattern. Understanding plant anatomy isn't just academic. It's essential for plant identification in the field, understanding how plants adapt to their environments, and has practical applications in agriculture and forestry. When you understand the internal structure, you understand how the plant functions and can better predict how it will respond to different conditions.
Meet Iyal, your AI study companion. Transform PDFs into smart study guides, practice tests and flashcards for UPSC, NEET, banking and any competitive exam.
Master your exam preparation with Iyal.